would like to show you a bit a lovely story we've been working on involving very old-fashioned science together with more modern science, and how we managed to make it, to complete it, to make a successful story. So just as a brief introduction, my group works on understanding gene regulation in the context of the chromatin. So on the top of the slide here, you have this rosary representing all the nucleosome at the chromatin state, and genes are regulated by two types of regulatory elements: the proximal regulatory elements called promoters and the remote regulatory elements called enhancers. I define enhancers as a remote control, simply influencing the activity of downstream promoters. And I say the activity of promoters, yeah, I will tell you in a minute why this word is very important. So enhancers can be located very close to the gene they're regulated, sometimes just 1 to 5 kb, but sometimes very far away, up to 1 being a base, so they can jump different transcription units to find a specific target promoter. So over the past probably 20 years now, my input in the field was to show that enhancers are key for many aspects. So they are key for the recruitment of the POL2 transcription machinery at the target promoters. So enhancers are definitely required for the recruitment of the polymerase to the promoters. Usually you have many enhancers regulating one gene. So those enhancers communicate to each other through chromosomal loop structure. In some cases, promoters can be covered by repressive complexes to block transcription, and enhancers are also important for the removal of those repressive complexes. It's the case for the polycom group repressive complexes. And when I mentioned to influence the activity of a promoter, is not only increasing the rate of transcription, but also the probability of transcription. So these are based on single cell studies. So the model we're working on is the hematopoietic system, simply because it's a very well established model. We can isolate many cells at different stages of hematopoiesis, from hematopoietic stem cells to the fully differentiated cells. And of course, it's a liquid tissue, so it's easily accessible. You can even bleed some colleagues to find different aspects of cell differentiation, something you cannot do with brain tissue, for example. So what we're trying to understand is the role of key transcription factors and epigenetic regulators for this process. So the key question here is to understand the lineage specification and the terminal differentiation. So we've been working on different aspects of hematopoiesis. For example, my PhD student at the moment, Camilla, is working on an epigenetic regulators influencing this particular lineage here in monocytes. But today I'm going to concentrate on the erythroid lineage and studying a specific transcription factor we've been working on. So erythropoiesis is a very attractive model simply because, it's the formation of red blood cells. So the cells simply get red because they get hemoglobinized. Hemoglobin is a tetramer of two types of proteins, α-globin and beta-globins. And the expression of this gene occurs very late during the process of differentiation in the erythroblast stage. And the expression goes up to the roof simply because the entire nucleus is focused on the production of globins to make those enucleated red blood cells. So you can find different, probably several millions of alpha and beta globin proteins in each erythroblast. So to produce hemoglobin, you need a lot of different transcription factors. So there's two sets of transcription factors. The first set is transcription factors that are tissue specific. You can see that these transcription factors are expressed at different stages of differentiation. And a key example is the GATA2, GATA1 switch. So very early, GATA2 is a master regulator opening chromatin at that stage. And when GATA2 has opened chromatin of target genes and enhancers, GATA2 will regulate the expression of GATA1. And in return, GATA1 will repress GATA2. So there's a switch between GATA2 to GATA1. And much later during erythropoiesis, a key transcription factors called KLF1 or eKLF or erythroid-specific KLF will make the final decision between megakaryocytes and erythroblast and will push the cells to the erythroid lineage. So the second set of transcription factors are ubiquitously expressed transcription factors, but these are very important for making a good connection between the tissue-specific transcription factors and the general Pol-II machinery. Many of these are zinc-finger proteins from the KLF group or SP-XKLF group, including ZBP89, but also other proteins like NFY binding catbox, and I'll go back to that in a few seconds. So the key things we've been analyzing in the past, and that's back to my postdoc back in the day, is to find when and where these transcription factors bind during the process of cell differentiation. So again, the beauty of this model is that we can either isolate primary cells, or we have different cell lines representing different stages of differentiation, and you can really study the dynamic of changes in one locus at the time. Of course, these days we do that by chip sequencing, but back in the day we did that by chip qPCR on just one locus, and I will summarize what we found. So looking at transcription factor dynamics, so again, this is the different progress of the transition between stem cells to erythroid cells. And this is a cartoon representing the α-globin locus. So this is 100 kb of genomic DNA. I didn't put any stones over here to simplify a bit the cartoon. So the first thing to know is that there are two genes in this locus. The first gene is an embryonic gene, the zeta gene, which is not expressed in adult cells. And then the two alpha genes. So these are two genes identical, 99% identical. And these are covered by CpG islands. And CpG islands has been shown to recruit repressive complexes as well. So in non-erythroid cells, the gene is repressed by polycomb group proteins. But now, if we move common myeloid progenitors, so these cells can be can form either megakaryocytes or erythroblasts, we have priming of one enhancers called HS-40. And that's really something you find in most of genes, is that enhancers always primed before promoters. And at that stage, you see the purple transcription factor here is actually GATA2, and GATA2 will be subsequently replaced by GATA1. So now if we move to pro-erythroblasts stage, when the cells are really engaged in the erythroid compartment, we have additional hypersensitive sites, so HS-10, HS-48. The numbers here represent the distance they have compared to these first genes here, but it's about 70 kb between these enhancers and the target genes. And at that particular stage, the promoter is cleared from these repressive complexes, and the promoter obviously becomes accessible to transcription factor. And the first transcription factor that comes in is NFY. NFY has a nucleosome type of structure and could be simply replacing the nucleosome there to make a nucleosome-free region. Then if we move to erythroblast, we have an additional hypersensitive site here, HS-33. I forgot to mention the purple transcription factor now is great. This is GATA1 engaged because of the GATA switch. And very late, as I explained earlier, we have a key erythroid transcription factor involved is EKLF, or KLF1. And we also found by doing looping assays that those enhancers communicate to each other by the formation of those mini loops. So independent of that, other things happening at the promoter level, we have additional transcription factor binding on those GC-rich box sequences, such as SP1 and SP3. Then the key other thing we found is that enhancers are also recruiting the polymerase. Now, because of interaction between enhancers and promoter, people may argue that the polymerase found that enhancers could be due because it's interacting with the promoter, and when you do a chromatin immunoprecipitation, you do cross-link the complex altogether. But we managed to prove that this is not true. And the beauty of the work was to having materials from patients where we had either the whole enhancer block here removed or the whole promoter and genes removed. And what we found is that when the promoter is completely absent, the polymerase is still capable to be recruited to the enhancers, but the other round is not true. So if the enhancers are missing, the polymerase never gets recruited to the promoter. That strongly suggests that the enhancers recruit the polymerase first, and then subsequently, boom, this will be transferred to the target promoter. So the work I want to present here is a bit of a follow-up of this, where we've been revisiting the alpha-globin promoter. to try to find more transcription factor binding there. So before I introduce this, I would like to go back to something maybe basic, and that's a review we published with Wendy Bickmore here in Edinburgh a few years ago. And I really want to advertise this beautiful review we wrote, not because I just want to advertise myself, but because this review is unique. And I'll tell you why it's unique is because I spent 2 weeks to calculate the precise size of all those nucleosomes compared to those big complexes of polymerase, mediator, and general transcription factors to see how that looks like at the proper scale. And usually people like to show that nucleosomes are those massive rocks in front of the polymerase, and the polymerase needs to shuffle those rocks to engage elongation. This is actually not true. Nucleosomes are really small compared to the Pol2 machinery and are the similar size to transcription factors. So although that was 6 years ago, please do read this review. I'm sure you're going to learn a lot of different aspects. But the key point I would like to make with this slide is that when you have a nucleosome-free region covering here the promoter, enhancers usually cover more than one nucleosome, sometimes two. A nucleosome-free region is about 200 base pairs, and 200 base pairs is actually a lot of space for transcription factor to bind. So it's something to keep in mind. So a colleague of mine, David Garrick, who is now based in Paris, has been reanalyzing the structure of the alpha-globin promoter. So these correspond to 200 base pairs of the promoter. The little arrow here represents the transcription start site. We have non-conserved Tata box here and a CAD box binding an FY. This has been already described well in the literature. And he's been focusing in these two GC-rich boxes over here that are very similar and looking at the transcription factor binding to this sequence. So back in the day, I'm talking about more than 20 years ago, and my entire PhD was based on the same type of essay, was to do gel shift experiment. Now, probably the young generation here in the audience may not know what is a gel shift. And I would be in the auditorium, I will ask people to raise their hand to see who doesn't know what is a gel shift. But now, thinking with this collaborate setting, you can actually raise your hands because you can ask questions as well. So to see who is not shy to click the button, raise your hands, and tell me if you don't know what is a gel shift. I'm on a full screen, so I'm not. I cannot really see if people will raise their hands. But Stephen, you may tell me if there's at least five. I can hear some noise. That's good. Okay, so I will explain this. The principle, it's very easy. It's an in vitro assay where you generate a lot of nuclear extract of the cells you want to analyze. And then you take this small piece of DNA, maybe, I don't know, 20 base pairs, and you label this radioactively. So you mix this piece of double-strand DNA with nuclear extract, and if the protein binds to your DNA, you will have a complex. So this is how it looks like on a gel. You have a polyacrylamide gel. The probe will migrate at the bottom of your gel here. It is very radioactive. There was a day we were doing this. I did thousands of gels when doing my PhD. But if you mix with a nuclear extract, these are B lymphocytes, you can see different bands. So that means different proteins here binding this sequence. And you can do that with different cell types. You cannot do competition with either the same probe in excess 100-fold cold, so non-radioactive probes, or different mutation, and you can then study the binding of the transcription factors. This sounds old-fashioned science, but you will see the beauty of doing those experiments to find something interesting. So this is with the probe 1112. You can see here the two sequences are very, very similar. And we found 3 different complexes in B cells and 4 complexes in K562, which is kind of an erythroid immortalized cell line. So when David did the same with the other probe, he found exactly the same profile, but the complex here is a bit stronger here. So try to investigate what is this complex number 4. So the additional experiment you can do, if you have a clue or you suspect some proteins to be the candidates, you can use antibodies and mixing this with your nuclear extract. And if the antibody recognizes this complex, you will increase the size of the complex, and then you will super shift those complex, or the band may simply disappear because the antibody recognized the DNA binding domain. So this is what he did. He used different antibodies, again, different SP, XKLF transcription factors known to bind those GC-rich sequences. He did SP1, and you can clearly see we have an additional spot here of SP1 on the top here. Then SP3, in this case, the band disappeared, and then KLF3, also called BA-KLF, where the band disappeared. So still, we try to find the extra band. E-KLF or KLF1 I mentioned earlier could be a candidate, but the antibody didn't react. And also we know that in K562, KLF1 is almost not expressed. So KLF1 is not the candidate. So still trying to find out what is this complex number 4 over here. So then they've decided to produce industrial level of nuclear extract and simply doing a chromatography, purification chromatography. And that was done in collaboration with Matthias Mann at the Max Planck Institute, who did mass spec to identify the candidate protein. We found 50 proteins, and the one that came on the top of the list was MAZ. So what is MAZ? MAZ means Myc-associated zinc-finger protein. It is an ubiquitously transcribed protein. It binds DNA through a GC-rich sequence, so G3 motifs, and also G4 quadruplex structure. It is regulated by phosphorylation. You can see there's 5 phosphorylation sites on the top here. Most of the time, phosphorylation increases the binding activity of MAZ. And fairly recent papers show that knockouts of MAZ is lethal, so the embryo dies around birth. So this is the gene protein structure, just to show that MAZ is a fairly short gene, has 5 different exons. Compared to the other SP XKLF transcription factor, it has 6 zinc finger instead of 3. So MAZ has probably a better specificity of DNA binding compared to the other transcription factors. And something to keep in mind, and I'm sure the bioinformatician will be pleased to see that, there are a lot of different MAZ variants, and that's super important when you think about targeting a gene. You have to make sure you target an exon that is common between all the different variants, which makes the story a bit more complicated, but not too much for this part of the work. So then we had to validate if MAZ was indeed the right candidate. So we did 5 different ways to validate this. The first way to validate it was to knock down MAZ, to remove MAZ from the nuclear extract. So at the time we did siRNA to deplete most of MAZ here in K562 cells, and you can see that indeed the complex D has disappeared. Then the second validation was to do the opposite experiment. It's using a cell line that where MAZ is not expressed at very high level, and then we did overexpress MAZ in those COS cells. You can see a big spot over here, and again the complex is now back here. So this is obviously still in vitro, and then the key experiment to do in vivo was to look if MAZ is definitely binding the chromatinized template using CHIP qPCR. So on the top of the slide, you can see the entire alpha-globin locus. This is 100 kb of genomic DNA. The announcers are located in the intron of this neighbor gene, which is a housekeeping gene. And you have the 2 alpha genes here. And over here, these are all the different TACMAN probes we've been designing across the entire locus. And these are the data, the enrichment of MAZ bound here. You can see that the highest peak is the promoter of the alpha-globin over here. But we also find a bit of enrichment of MAZ at those key regulatory elements of alpha-globin. So far, so good. Everything looks really beautiful. So then we did more functional assays, and the first one to look if MAZ downregulation would affect alpha-globin expression. So we've been using, again, K562 cells that express mostly alpha-globin and not the embryonic globins. And here we've been using 2 shRNA covering this particular exon that is common with all the different MAZ variants. And the work was beautifully done by the lab of Jan Frins in Bristol. right in the middle of the pandemic. So I can tell you these all aspects being extremely stressful for us. And you can see that strong downregulation of MAZ is affecting the expression of alpha-globin in these cells. The downregulation is obviously modest because keep in mind MAZ is not the main regulator of alpha-globin. Obviously, there are many regulators. So removing one transcription factor only moderately reduced the expression. So this is in a cell line. And of course, the other functional assay would be to look in primary cells. So this is a bit more sophisticated experiment because you need to proceed with a differentiation assay and to target the cells at early stage using a lentivirus. So we've been infecting progenitors here called birth-forming unit erythroid and keeping the cells for 15 days in culture. So the differentiation markers we're using here is CD71, which is the transferrin receptor expressed at the intermediate stage over here, and then the glycophorin A, which is expressed at a much later stage. So in a normal situation, cells will start accumulating these markers, and then the fully differentiated one, the GPA, which is what you can see here on the flow cytometry. These are the untransduced cells of the scramble vectors. You can see the cells reach a fully differentiated stage, but with the 2 shRNA, you can see a drop of final differentiated cells with an accumulation of progenitors. So on the middle panel, this is just the quantification of the flow cytometry. You can see an increase of the progenitor pool and a decrease of the fully differentiated pool. And of course, the beauty of working with red blood cells is that you can simply appreciate easy physical phenotype is that the cells themselves look anemic and the red pellets become pale. So the cells simply don't differentiate. So because the cells don't differentiate, so they're still blocked in this kind of stage, obviously anything which is downstream will be affected. And that was confirmed by qPCR looking at the RNA level of both alpha and beta globins. You can see a drop of the two genes over here, but also at the protein levels. So this is all very good. And then, of course, many transcription factors involved in erythropoiesis have been found to be involved in disease affecting alpha or beta globin expression. So these are the key features of thalassemia. So thankfully, because of the beautiful work of one of our scientists here in Roslin, Albert Teneza, generated this gene atlas database. And Dasha, my postdoc, has been looking at gene atlas, and we found actually 3 different variants of MAZ associated with erythroid traits, which really emphasize further the involvement of MAZ in erythropoiesis. So thanks again to Albert for this beautiful resource. So then, of course, we had data with chip qPCR. So we've been sending this material for sequencing. We could look at the entire genome. So again, we reconfirm our previous data showing by chip sequencing that strongest peak are found on the alpha-globin promoters, but also a peak at the alpha-globin regulatory element, HS-40, called multi-conserved species regulatory 2 elements over here, and almost nothing at the beta globin locus, although we have those very tiny peaks who did pass the peak calling, but we suspect this could be a protein-protein interaction rather than direct binding of the DNA. So now go beyond globins. Now we have obviously the entire genome to look at. This is what we found. And again, the work, everything was done from this part by Dasha Dean, who was my postdoc in Roslin, who left quite recently. So most of MAZ peak, we found in total 10,000 peaks, and most of peaks are found at the promoter regions. But then the second big class of region where you can find MAZ are intergenic region or intronic regions, where most of the time are also enhancers. So these are the 2 main regions where we found MAZ peaks. And then by collecting all those peaks, you can do a motif analysis and using MEME. And what we found is a MAZ bind mostly a G3, G4 motif, and not G3, the G3 as it was previously explained in the literature. So this is the motif we found. So G3, G4. Unfortunately, we don't have the 5G for you guys, but that's what we found in most of peaks. And then we've been looking at MAZ signal compared to other key genomic features. We've been looking at open chromatin region by ATAC-seq. You can see a correlation between MAZ signal and open region of the chromatin, a correlation with trimethyl K4 marking promoters, and also acetylation of lysine 27, which marks both enhancers and active promoters. Correlation with the polymerase, and inverse correlation with the repressive mark, which is the mark deposited by the polycomb group protein I mentioned a bit earlier. And something not included in the paper is a correlation with CTCF. There are 2 papers that should be online very soon, showing that MAZ also interact with CTCF in a subpopulation of peaks as well. So MAZ could also have a role in the global chromatin architecture. So then Dasha looked at some ENCODE data, and ENCODE thankfully did some chip sequencing with MAZ in other cell types. And you have here, these are hepatocytes, lung cells, B cells, and breast cancer cells. And I forgot which one is the last one here. And she has defined 2 types of peaks, those called the common peaks. Common peaks are peaks that are in common at least between 2 different cell types. And then the erythroid-specific peaks that only specific in erythroid lineage, and these are the one here at the bottom. So that's 17% of the peaks. And we found that all those peaks are located in key erythroid genes, perfectly fitting our previous data showing that MAZ is important for erythropoiesis. So to push this a little bit further, she's been looking at the proportion of peaks found in promoters versus enhancers. And something interesting she found is that in the common peaks, apparently most of MAZ peaks are found at promoter region and much less to enhancers. But in the erythroid-specific pool, it's the opposite situation where most of peaks are found in the regulatory elements. So it appears that MAZ is very important at enhancers regulatory region in erythroid lineage, emphasizing more its role as a regulatory protein rather than just housekeeping transcription factor binding just to promoters. So this is a correlation we found with different genomic features at the non-TSS set. So they are obviously stronger MAZ enrichment. The peaks are stronger in the erythroid set. We have a stronger correlation with open chromatin region again, and a strong correlation with monomethyl K4, which is specific of enhancers. We don't find these at promoters. And indeed, the mark of promoters, there's a reverse correlation with MAZ binding. So that strongly suggests that MAZ is important for enhancers activities in red blood cells. So then the last thing that Dasha has been looking at is to make any correlation between MAZ and a key master regulator in erythroid cells. And I mentioned GATA1 earlier on. So what she found is that if you look MAZ at promoters occupied by GATA1, there's a correlation at the promoter, but this correlation is even stronger at the enhancers over here. And the reverse situation is also true. If you look at GATA in the MAZ pool, we saw a correlation at the promoters, but the correlation was even stronger at the enhancers pool as well. So up to 86%. So the work was actually accepted recently and still not online at the moment, but is being accepted in Blood Advance. So in conclusion, we use an unbiased approach leading to the identification of MAZ as a transcription factor controlling the erythroid program, originally by looking at MAZ binding to the alpha-globin promoter in vitro and in vivo, and we found that it's definitely required for erythropoiesis. Clinical significance, we found MAZ variant associated with erythroid clinical traits. That's because of the Gene Atlas database we've been looking at. And by chip sequencing, we found that actually many erythroid genes are regulated by MAZ, and we found an association with GATA1 binding at those enhancers. So now how MAZ regulate erythropoiesis, and that's where the model gets a bit complicated, because we found that actually MAZ regulates, as I said earlier, erythroid genes. So GATA1 gene itself and KLF1 genes are occupied by MAZ, and this is an example of the chip sequencing we found. This is the KLF1 promoter. And what is also complicated is all those KLF protein regulate the expression of each other. So to understand why different protein can compete for the same binding sequence based on the gel shift we found earlier, understanding what's the hierarchy and at what time these protein compete with each other is something we need to find out in the future. With this, I will stop and thanks all the people involved in the work. So Dasha Dean, my postdoc, who's been completing this beautiful paper, and Dasha is now a senior bioinformatician at the University of Newcastle. David Garrick at the University of Paris Diderot, who initiated the work. Matthias Mann and Falk Butter at the Max Planck for the MAZ Spec aspect, and all the primary erythroid cells were completed by Jean Frein and her team in the middle of the pandemic last year. And obviously my former lab where David Garrick initiated the work, and Michelle Holland, who gave a talk a few months ago over here, who was also involved in this. And there's this beautiful Rosalind in May last year, right in the middle of the lockdown. So when it was completely empty, and I keep advertising to people, says the bad weather in Scotland is not what you think. And this is an average picture you can find every day when you come over. So thank you very much for your attention, and I'm happy to any question, obviously. Thank you.